You are welcome to my YouTube channel. The topic to be discussed is the lectin pathway of the complement system. The complement system is an integral part of the non-specific immunity or innate immunity, which helps to eliminate invading pathogens. It is important to note that there are three pathways of activating the complement system. The classical pathway, which has been discussed in another video, then the lectin pathway, and the alternative pathway. Now, the lectin pathway. Here you have lectins in blood circulation, and these lectins are produced by the liver. What are they? They are proteins that recognize and bind to specific carbohydrate groups. These specific carbohydrate groups can be found in bacterial cell wall, fungal cell wall, protozoal cell membrane, and the viruses. The first one to be discussed is mannose binding lectin. It has a similar structure and role with the C1 component involved in classical pathway. But this mannose binding lectin binds specifically to mannose in these microorganisms listed. Now you also have a group of compounds known as ficolins, similar to mannose binding lectin. There are three types. You have ficolin 1, ficolin 2, ficolin 3. These ficolins are known to bind to acylated sugar residues such as N-acetylglucosamine, which is found in the peptidoglycan polymer of the bacterial cell wall. Now, the mannose binding lectin and other ficolins are known to bind to MBL-associated serine proteases. What are they? They are known as MAPS1 and MAPS2. MAPS1 is the first to bind to the immune complex, and then followed by MAPS2, which cleave intact C4 into C4a and C4b, and then the C4b, which is the larger fragment, initiates the cleaving of C2 into C2a and C2b. The C2b binds to the immune complex, giving C2, giving C4b and C2b, which is known as the C3 convertase. This complex is able to cleave C3 into C3a and C3b fragments, and the C3b will combine with the immune complex on the surface of these microorganisms and initiate the cleaving of C5 into C5a and C5b. That is, the immune complex having the C3b is known to cleave C5 into C5a and C5b. Now the C5b combines with the immune complex, and then this leads to the combination of C6, C7, C8, and C9 components, leading to the opsonization of pathogens. Now, it is also important to explain what opsonization is. The components that have bound to the surface of the microbial cell makes them more attractive to neutrophils and macrophages for phagocytosis. It is important to note again that mannose binding protein is an opsonine, and it binds to several soluble immune complexes and microbial pathogens. Here you have the diagram showing the activation of C3 and other components by the mannose binding lectin-associated serine proteases, which can be seen below. You have the MBL plus the serine proteases, the MAPS1 and MAPS2 bind together, which following that, then C4, C2, and C3 are cleaved and bind sequentially to the immune complex, generating C5 convertase, that is C4b, 2b, 3b. These will now activate the C5 component, which is cleaved into C5a and C5b. C5b will combine with the immune complex, followed rapidly by C6, C7, C8, C9, and these leads to opsonization of the pathogen. What are the functions of the lectin pathway? One, opsonization, which I've already explained, and then elimination of immune complexes. Thank you for listening.